Hey everyone out there, my name is Kaitlin Bryson and I am going to be talking today about mycology and art, and we're going to do a quick workshop, hopefully if we have time. If I run out of time, I will be posting some videos about the workshop so you can follow along. For the workshop, there's a few materials that you need, and if you don't have those things, such as a bag of mycelium, you might want to get those things first, or you can start practicing cultivation, mycological cultivation, on your own with some other practices that I'll talk about later. But first, I wanted to give you a introduction about mycology and about myself, and so I'm going to start there. So let me. All right. So this talk is called Mycelial Talk and Workshop, I should say, Mycelial Landscapes, How Fungi Shape the World and Take Shape of the Future. And once again, my name is Kaitlin Bryson, and I'm an artist, mycologist. I work at the UCLA ArtSci Center, and I also am an educator teaching remotely at University at Buffalo in New York. So, fungi. Fungi are, let me get situated here. Fungi are ubiquitous queer organisms in our environment that create and facilitate the world around and within. This unique queendom on the tree of life blurs the boundaries of worlds defined by humans. Often they exist and thrive within the fringes, between life and death, between the singular and the multiple, between sexes and identities, between taxonomically distinguished, in harsh and impossible environments, and truly beyond our understanding of time. Fungi are permeable, promiscuous, and profound. Today I'm going to share with you my knowledge of fungi, which, in comparison to their breadth, is microscopic. I will talk about what I have learned through working and collaborating with these organisms as an artist and an activist, sharing what I believe to be vital information to us humans in the wake of our environmental, social, and political disruption. As I come to you today from smoky California in the midst of this unsettling display of climate crisis, it feels fitting and appropriate to talk with you about fungi, and consequently about decay, metabolization, and regeneration. I've also prepared a simple workshop for you to follow along with anytime. And as I noted, you have to find yourself some mycelium first. Materials for this workshop will be posted in our chat on YouTube and linked in a Google Doc, so you can always follow along. And again, once I have the videos up, if we don't get to it today, then you can access it at any time. But what we're going to be looking at in this workshop are making mycelial hands to show how fungi take the shape of their environment, and also to propose a metaphor for human collaboration with them. What if we can reach out and touch each other? Does this change how we relate to and interact with them? The more than human world? How can we care for ourselves? These are some of the things that we're going to be thinking about and talking about today. So I became interested in mycology about 7 years ago when I was working and studying as a permaculture farmer. I was introduced to the fungal world by the way of the sort of OG of mycology, everyone's kind of introduction, Paul Stamets, whose book Mycelium Running was floating around a farm that I worked on at the time. I was deeply interested in systems at the time, and especially fascinated with how desertified ecosystems could be renewed by growing food and implementing more dynamic and layered systems with plants. The foundation of all these systems is the soil, and this is truly where my obsession with fungi began and grew from. I learned about the mycorrhizal sphere and soil communities. I learned about decay and nutrient distribution, and I learned about a process called mycoremediation. A couple of years later, I started a graduate program at, called Art and Ecology at the University of New Mexico. And I went into this program with this desire to apply these sort of grand ideas of mycoremediation to landscapes suffering from toxic contamination left over from extractive industries such as mining. So this is a mine here on the right side of the screen that's in my home state of Nevada. I was living and working in Peru, and from these experiences, working on these farms and learning about indigenous farming practices, I thought about these similarities between these two systems, or between these two structures, excuse me, and I had this idea of what if we could turn these giant open pit mines into farms, sort of recreating the environment and also creating a giant living, breathing sculpture that would facilitate environmental and socioeconomic renewal, as well as community development in the soil and within the human communities. Although I was not able to even come close to that scale of remediation, I did, however, cultivate an art practice that works systematically with fungi and is geared towards processes of remediation. I studied in graduate school in both the art and biology departments, working with great mycologists such as Peter McCoy and Dr. Donald Napvig at UNM. And these experiences really helped my knowledge sort of grow, and I also got to really expand my fungal skills. Now as an artist, I work with issues of environmental and social justice through creating remediative living sculptures. I work primarily with fungi and mushrooms as a material, but also as collaborators. And I tend to work with fiber, installation, weaving, social practice, performance, kind of running the gamut, but all sort of geared towards these messages of the mycelium. Let's sort of start with situating fungi. So fungi are eukaryotic organisms that are nested between animals and plants on the tree of life. They've existed on our spaceship Earth for about 2 billion years and have played an absolutely integral role in the habitation of our planet. Fungi are not plants and actually are more closely related to humans. Some of the main distinctions that we can see or that we know is that fungi have cell walls that are made of chitin and not cellulose like plants. Fungi are heterotrophic like animals in that they acquire food by absorbing dissolved nutrients, and fungi do not photosynthesize like plants. The morphology of fungi, so it's sort of referring to their body and what their shape is. Their morphology can be defined by mycelium, and mycelium is this white feathery substance that you see here and that you've probably seen before. You might see it in the forest floor, like picking up some leaves, or it might be growing on an orange in your house. But this is sort of the overall like major characteristic of fungi, and most all fungi except for yeast form mycelium. Mycelium grows in decaying matter. It grows in plants and vascular systems and root systems. It grows on rocks. It grows in our bodies. It lives in our bodies, and it's sort of abounding. So mycelium is the vegetative body of the fungus. If we compare fungi to an apple tree, for instance, we can think of the mycelium as the trunk, the leaves, the root system, while the mushrooms, what you might be familiar with when you think of fungi, are actually like the apple. So they're sort of like the plants or the fruit. Mushrooms are responsible for bearing genetic information, so they fruit when they are releasing spores. And here in this image, this is a sort of here is what we call hyphae. It's a cross-section of a hyphal cell, and mycelial networks are made up of hypha, which is this one cell thick thread that grows three-dimensionally, multi-dimensionally throughout its home. So the actual shape of the mycelial body is best described by mycologist and author Merlin Sheldrake, who likens the body of fungi to water. Mycelium takes the shape of whatever it's contained within. So for fungi, their habitat is simultaneously their home and their food source. So they truly are what they eat. And I kind of say this over and over, but I want it to be kind of like embedded into you. This phenomenon of mycelial growth is one of the key reasons we can work with them to co-create materials. And we can shape fungi and they in turn shape us. And we kind of have this very interesting relationship. So for the ecology of fungi, their food source and lifestyles help us distinguish how to classify them, how to begin to classify them. However, as the field of mycology expands, it's becoming increasingly evident that their behaviors are queer. They don't always behave in categories. It seems that fungi are or contain a sort of encyclopedia of the Earth's history that's transcribed biochemically within their genomes that allow them to switch roles or functions given the environmental circumstances that they are presented. However, for the sake of satisfying our human brains, I'm going to talk about a few of the classifications of fungi that I tend to work with and find most important. So the last couple that I just showed you were the saprotrophic, the decomposers, and the symbionts as on this slide. So decomposers, obviously they live in decaying material. They consume whatever is sort of on the ground, or they consume rotting wood, whereas symbionts kind of live within another organism and they exchange nutrients. We're gonna start first with the ASCOMYCOTA, ascomycetes, thinking about molds, medicine, and compost. So ascomycetes make up 75% of the known fungal species. They can be recognized as mold yeast, as well as relationship-forming fungi, either mutually beneficial or pathogenic. They can be, a lot of the ascomycetes are edible species, and some are the ever sought after truffles. And ascomycetes generally grow very quickly and can produce sexually and asexually. The spores are everywhere. They're in your house, they're in your body, they're in your nose. And so they're on fruits. They are really everywhere. So this is why it's very sort of common to have them around. Yeasts are probably some of the ascomycetes that you might be familiar with in the process of fermentation. The yeast here, Saccharomyces cerevisiae, converts carbohydrates into carbon dioxide and produces alcohol. And then we have, of course, bread and cheese and wine and all the things that we love as humans. In terms of medicines or antibiotics, in 1928, we have an accidental discovery of penicillin made by Alexander Fleming that sort of just grew spontaneously on a culture of staphylococcus, staph infection that was growing on a petri dish. And there was a penicillium spore that landed in this petri dish while he was away for a couple weeks. And it started exuding out these metabolites that are actually right here that are defending the staph infection. But this was discovered in 1928 and obviously revolutionized modern medicine, but practiced throughout time with indigenous peoples. Molds would be put onto wounds to treat, to treat wounds and sores because of the antibacterial properties. So in my artwork, I have worked with ascomycetes mostly by accident. Through my process of cultivating mushrooms and sort of like learning how to grow these organisms, I would create these habitats that were very ripe for mold contamination, basically, or for anything to grow, anything that wants sugar and water and a sort of humid, delicious environment. So although in mushroom cultivation, contamination is understandably avoided at all costs, as an artist, I find it a most interesting teacher and a humbling parable about my role as an artist and a maker in this world. Mushroom cultivators are generally seeking to cultivate a single species, so they want to do everything that they can to get competitor species out of the way by means of pasteurization and sterilization, creating a really sort of hospitable environment for single species, usually a mushroom-forming species. So we go to these great lengths to create this sort of sterility. And since molds grow so fast, they often will outcompete those sort of targeted species that they're trying to grow. I, however, am just interested in sort of watching these things unfold, and also am inspired by and invited by the author Anna Singh to see contamination as collaboration, and really just question this idea of isolation and saying who can grow where, and especially when I'm the one creating the environment in which they're growing. So this is a piece that I was trying to grow oyster mushrooms, and instead there were over 6 detectable species of ascomycetes that were growing on this piece that I called numinous decay. Another thing that's really important is that these species often are, because they sort of grow so fast and they're really strong and hardy, they can start the process of decay really quickly, and they can initiate compost processes. So this piece eventually became part of my compost pile. The BASIDIOMYCOTA are perhaps the most recognizable species of fungi that you might know. To reproduce basidiomycetes make this tremendous morphological shift, condensing their mycelium into the fruiting bodies that we call mushrooms. And these organisms are saprotrophic, so again, they live off of decaying material and are actually some of the world's oldest organisms, and therefore are some of the most evolved creatures on our planet. One of the many notable things about these species is that saprotrophic fungi have learned over eons how to digest lignin, which is the very sort of difficult and complex chemical that makes up wood. And this very, very strong bond of wood is sort of broken open by these decaying fungi who are in search of cellulose and hemicelluloses. But in this process, they kind of go into this very complex carbonic chain, and they break open these chains by means of oxidation. And when this happens, it kind of like unlocks all these bonds, and a whole suite of other organisms can come and move in and begin to further decay the material. What's interesting about this is that this is actually one of the reasons why mushrooms can do what's called remediation or microremediation, because a lot of polycyclic hydrocarbons and plastics are based off of this this structure right here, this lignin structure, similar in sort of creating these polymer chains. So I have worked with various aspects of microremediation, again, inspired by some of the mycologists who have come before me, and have worked to create this, to create these sort of works that can remediate, but also to utilize these potentials in the actualization of remediation in the environment. So for my graduate thesis work, I was very fortunate to collaborate and partner with an amazing organization based out of Espanola, New Mexico, called Tewa Women United, who really work towards environmental justice issues in New Mexico, really kind of stemming from the development of the atomic bomb. And I worked with this organization and created these mushroom pillows, these pieces that were they were based on specific sites chosen for remediation, and they were given a specific fungal partner that was targeting this contaminant in a given location. And then eventually they were created and buried with the women of Table Women United and community in Española and Rio Arriba County. And this was kind of the first project that really unfolded and created a series of works that I'm still continuing to work on today, but also kind of unlock these potentials. And this project eventually evolved into being a real kind of social and environmental justice, I guess, larger life work with Table Women United, in that we were eventually, it kind of evolved into proposing a formalized mycoremediation project to Los Alamos National Laboratories for contamination issues that were still stemming from issues that were created by the labs, basically. So I was very lucky and humbled to stand alongside Table Women United in a public hearing, speaking out against the injustices from LANL, Los Alamos National Labs. Another thing about the sort of decaying fungi that I'm obsessed with can be kind of looked at closer in a most recent work that's called When Civilization Fails to Serve, It Must Collapse. This new work I've made this during the pandemic and the social and racial justice movements that are happening right now. And on this work is embroidered text that I've written that illuminates a call to action for the decay of these obviously collapsing and oppressive systems. It's further emphasized by the composition of Saprotrophic Pleurotus osteratus, which is consumed and sort of becomes the text, becomes what's written on the fabric sculpture. On the backside of the sculpture, inscribed in honor and prayer to all of the people who have been killed by the police since 2014. And these names are prayers and messages written to be consumed and therefore multiplied by the mycelium to really initiate this social change. And after this sculpture fruits, I'm going to bury it in a disclosed location in Los Angeles to initiate that change. So with the saprotrophic fungi, with the decayers, I think that not only are their potentials really amazing in what they do in terms of reclaiming and redistributing nutrients in the ground is really important, but also the metaphor that kind of provides and the potentials that come from both of those things working in tandem. The next category are the mycorrhizal fungi, who have become stars, and rightly so. So over 90% of the Earth's terrestrial life, the Earth's plant life form mycorrhizal relationships. This word mycorrhizal literally means root and fungus and refers to the root of a plant being colonized by a symbiotic fungus. So mycorrhizal fungi invade tree roots or plant roots and tap into their root systems and obtain nourishment from the host plant in exchange for excess carbon that the plant is producing through photosynthesis. So fungi have these incredibly dynamic, expansive systems as the mycelium's growing, their hyphal networks are expanding, and they can tap into minerals and immobilize these sort of minerals and nutrients and give them to the plant in exchange for this carbon that are usually otherwise unavailable to plants. So they also have been kind of found recently to be assisting and modifying plant behavior for the benefit of the multiplicity of the ecosystem and the species at large. So they're expansive and interconnected systems that, again, here are some images of these things in action. And I think that I'll post this in the chat or maybe in my video, but the mycologist Suzanne Simard who coined the term, the Wood Wide Web, has described this very, very well, and I'm obsessed with her and her work. But it's also been found that these networks are not only providing this amazing host of exchanges that are happening, but they're also specifying different species or different, they're specifying their kin, and they're focusing on giving nutrients to their kin. They really assist and modify plant behavior for sort of the whole ecosystem. And they're also communicating. So we think about this as a form of electrochemical communication. These organisms are sort of, because they're living, they produce electricity, and they're sending these signals all throughout the environment, kind of letting the ecosystem know what's going on. So not only are they bringing nutrients and minerals in and giving those to the plants, but they're also kind of, telling them what's up. So this, lately, the most recent work that I have been fortunate enough to make for this festival with my partner really talks about mycorrhizal networks and the importance of these communication methods. And we were interested in looking at some of, like, how could we think of portraying these signals? How could we talk about this importance between species, and especially when these things are microscopic and we can't really hear them, they're inaudible. So I was inspired by, and continue to be inspired by, Merlin Sheldrake, who through biosonification, so putting electrodes on a mycelial body, and then ran that into a MIDI controller, and then it produces a sort of an electric wave, and then that is transferred into a sound. And so what you're hearing is actually the electrical signals that the mycelium is emitting. And this, again, this produced this kind of desire to think about, like, what's being said, and could we begin to decode those messages? Should we decode those messages? And I think that we should. I think that it is about not necessarily decoding their messages into a language that sort of matches ours, but really just this idea that we need to listen to other species and to things outside of ourselves, especially when it comes to issues pertaining to the environment. So this piece entitled Transrelations, that's debuting tonight, that I worked on with my amazing partner, Matea Friend, and also another friend composed the audio, Ian Nelson. It's happening tonight at 7 p.m. I can share those links as well. But we were kind of thinking about how we could, again, bring this to life. And so here's like a little video of what we did. So through this material of weaving, actually weaving in between different species of trees at the UCLA Botanical Gardens, sort of using weaving as this ancient technology and mycelium as an ancient technology, and then projection mapping, using projection mapping to trace light and electrical signals and that are happening between the mycelium, and then producing a sort of soundtrack that goes with that. So again, it will be talked about more this evening at 7 p.m. But that's kind of the materiality and the thoughts that we are working with. I want to thank the people that I have learned from and that I really admire a lot. So starting with the fungi, Dr. Donald Natbig, Peter McCoy, Willoughby Arevalo, Suzanne Simmerd, and Merlin Sheldrake. So, sped through the talk so we could get to the workshop in which we are going to make mycelial hands. And I actually, I did this workshop with Willoughby Arevalo in Mexico at a residency, and it's a really great way to see sort of what happens when fungi grow and consume a substrate and how that then can become a architectural material that's very rigid and strong. And there are amazing companies that are using this materiality. There's one called Ecovative, who I recommend purchasing mycelium from if you're going to do the workshop, and MyCoWorks. There's also a lot of architecture that's being built in tandem because you can use this material, hold it up, as a building material. It's super strong and dense and has a lot of potential also, especially for insulation and building materials. So I'm gonna pause my screen share right now and move to a different location. That's set up. I'm also, if anyone has any questions, they can be filtered through the chat and in Zoom here while I move to a different place. Yeah, that'd be great. Thank you. That would be awesome if you can.
Hi everybody, this is John. I'm going to be stepping in as kind of the host for now, and I'm going to pass on some questions to Kaitlin. It looks like we do have a question from Christoph in the chat, and for Kaitlin it says, in your work, are you more touched by the similarity between fungi and humans on a biological or rather symbolic level, or is it the interweaving of both?
Hi, Christoph. Thank you. It's definitely both. I think that initially I was really inspired by the metaphors that I was learning about and reading about, like back in the early in 2013 when I was farming and kind of got into it. But then once I started learning about the science and sort of the actuality of what was happening and breaking that down, it really became like this huge epiphany of the actual symbiosis between those things. And that's why I've studied art and science together, because I think those things really feed each other and create an incredible sort of like networking community that can be actualized. Also, like learning more and more about fungi, like over the past 7 years, I've just gotten so obsessed. And like the more that I find out or learn about their lifestyles and their behaviors and their sort of like, their queer way of being, the more I like those sort of the scientific attributes or their biochemical attributes become also metaphors themselves. So it's just this constant process that is evolving together, I would say.
We also have a question from Ashley. It says, of your projects, which has proved to be the most successful and why?
I would think that, well, success is an interesting word, but the most profound project and really the most profound relationship that I've had in my in my life and in my sort of art practice was working with Tewa Women United in New Mexico. And because of these really deep relationships that were formed and just the incredible work that we did sort of together to fight back and help resolve or talk about resolving some of the issues that really continue to permeate their world. A lot of lands all throughout America. And so in terms of the scientific remediation aspect of that, that project was not successful. We weren't able to really get quantitative data that showed us that we were in fact remediating. The projects were done on a small scale, so it was really hard to test. And also it was kind of impossible to gauge the actual sort of success, the scientific success of that project. But yeah, in terms of what the art was doing and the community that was formed, the sort of like radical network of people that came together to do a lot of community projects that kind of were part of that whole work. We had workshops, we hosted workshops, we did DIY mushroom cultivation, we buried a bunch of things together, we had ceremonies, and all of that was incredibly successful and very profound.
Great, thank you. I think that's good for questions right now, so we can get into the actual workshop part of it. And awesome. For those in the chat, if you have more questions, you can post them, and if we have time at the end, we'll get to them.
Great. So I'm here in my kitchen. I have set up, sorry, I don't have a webcam situation better, but I have a simple little table set up with my materials. I have some gloves, paper towels, sterilization stuff, rubbing alcohol, AKA. Scissors, tape, flour, water, 3 cups of water, and my substrate that I got from Ecovative. Ecovative is an amazing company that is based out of New York, and they're providing these bags of mycelium that's dormant, and you can activate them. It's really simple, actually. They kind of cut out this huge aspect of the cultivation process that is really lengthy. And if you're interested in doing that, I highly recommend you do it because it's very important to sort of know the foundation of that. I learned how to cultivate through the Radical Mycology platform and through Peter McCoy in particular. And that was super helpful, but I also love that I can purchase ready sort of mycelium that I can activate and work with a lot quicker. I do have 2 books that I'll also post in the chat that I recommend if you're interested in cultivation. But for now, I'm going to show you really quickly this 2-part workshop. It has to be done sort of 5 days apart. So the first thing that you do is you activate your bag of mycelium. So we're going to start with that. John, can you hear me okay? So the first and foremost thing about any aspect of the mycological process is that you do want things to be as clean as possible. So I have already sterilized and kind of cleaned this table, but for the sake of best practices, I'm going to do it again. I'm going to first grab some gloves and put them on. Actually, first. My hands are clean, but I will clean them again. Arms, my jewelry. About there. Okay, now gloves. And actually, during this time, if there are more questions, it would be good to ask them now while I'm kind of in this process. So again, I'm going to sterilize first. I wiped this down with disinfectant wipes before, and I also sterilized each individual thing, like the outside parts of everything. So I'm going to just do that again with the rubbing alcohol, using a lot of paper towels, and I've got my scissors, which are important to also clean. Okay, so I got this bag of mycelium. It has a filter patch on it. One thing I didn't mention in my ecology portion of my talk, because I was feeling nervous, was that mushrooms respirate like we do. So they need oxygen, or mycelium and fungi respirate like we do. So it needs oxygen. So this bag has a patch for respiration to take place. So to wake this up, I'm going to cut below the seal. And I'm going to stand back because I don't want to breathe in it. Actually, for that, I'm going to take my jar of water, 3 cups of water. Again, this will all be posted. And I'm going to put in 4 tablespoons of flour. And again, I'm going to always stand back because I don't want to breathe into it. Now, opening my bag, standing back, I'm going to pinch both sides and pack it down. This is a bag of reishi mycelium. Reishi is an amazing mushroom and very medicinal. It's an adaptogen. It's super good for your body, but it also has amazing mycelium to become sort of rubbery and can be used as a really great building material. So I have my jar of water and my flour, and I'm just going to pour it in. Okay. Squeeze out the excess air, and I'm going to tape off my bag. But I don't want to cover up this patch because it's really important that it's round, that it stays open. I'm just going to leave that. And really quick, I have some tape that I pre-cut, but it looks like it fell off my table. So just touching only the tape, I'm taping my bag to the outside. If you try to do this with gloves on, the tape most likely will get stuck to your gloves. So it's kind of actually necessary to take them off. All right. So now simply have this bag of mycelium with the top all taped up. And now we have to get that flour all mixed in with our substrate, which is hemp. So pick it up. I'm gonna take a little more. Leaking a little bit, but okay. And now this will sit for 5 days, and it will start to myceliate and become white and beautiful. What's also really cool about Ecovative is that they're using a bunch of bio-waste. They're using hemp bio-waste to actually create these building materials. And so, again, they're closing this loop, this waste stream, by growing materials on bio-waste that happens from agriculture and allowing us to learn about mycelium and grow with them, but also to create packaging, to create insulation, and just all kinds of materials. So now I have a pack of mycelium that I have already started. So let's start afresh with cleaning things. I'm gonna put this stuff away. Are there any other questions?
We had a comment from Christoph said it's kind of ironic that as mold, it moves where it shouldn't, and then at the same time it's so sensitive that you have to clean your whole house and create a kind of artificial environment for them just to be able to grow on your own.
Yeah. Could you say the first part of the comment again, John?
Yeah, so it's kind of ironic that as mold, they move where they shouldn't. So they're sort of, you're fighting against the spread, but simultaneously, if you want to actually cultivate, you have to really clean everything.
Yeah, this is what I find also, yeah, very interesting. Is that like also in the environment, if these reishi mycelium or these reishi mushrooms were growing in their own environment, they wouldn't need such sterilization, right? Because they're just around like a whole like billions of other microorganisms that they can totally do their thing and defend as needed. But when we isolate them, we sort of this is this tension that I'm interested in again, like what happens when we bring things inside and we isolate them, then all of a sudden we have to like hyper isolate them. We have to obsess over sterilization, over creating these habitats that nothing else can live in, but everything wants to live in those habitats because there's sugar, there's humidity. So it's super ironic, and I find it very interesting. And also, yeah, Christoph, another concept that I concept for art making that I think is very fascinating and kind of like there's no real sort of like landing on it one way or another. It's just kind of interesting because it is what it is. Okay. This doesn't need to be, I usually put it in kind of a very like quiet enclosed place, like a cupboard maybe, or somewhere where it's not going to get disturbed. It doesn't love sunlight, but it's also okay if there's some sun hitting it. But I would recommend that it goes in like a cupboard. You don't want it to dry out. It's very important that it kind of just like gets to fully grow and consume the substrate. So I'm going to put it over here. Grabbing my other bag of mycelium. You can kind of see some at the bottom. I put it in the fridge so it would go dormant for a little bit. But now I'm going to actually re-clean everything, and I'm going to start with my hands. Any more questions? All right. Cleaning the surface. Okay. Now, actually, first I'm going to open my bag. So I've used some of this mycelium for the demonstration, which I'll show you in a minute. Since this is going to be the last time I use this mycelium and it's consumed as substrate's really healthy and strong, I'm going to put gloves on, but I'm not as worried about it getting contaminated. Thank you. Almost done. It's a huge rush. I really rushed through my talk too because I was trying to get to this part. Okay, so I've got the gloves, and all I'm gonna do is take this mycelium and its substrate, and I'm gonna put it into the gloves. So I'm gonna wake up the glove and just go for it. Making a gigantic mess. In this process, this starts to happen where it gets all caught up in the fingertips of the glove, and all you have to do is just pinch it at the top like a water balloon, and then kind of squeeze it into the fingers. It smells really good, but I'm making a gigantic mess. This mycelium is also really good for compost. So if you have some excess like this that I probably won't use because or any other gloves or other building projects that I'm doing, I just put it in my garden. And then you tie it off. Make sure you don't put too much in it, like I just did. All right, and you've got your hands. Since mushrooms, these mushrooms and fungi respirate like we do, we need to cut little holes in the glove. And if you let your glove fully myceliate and grow out, mushrooms will most likely fruit out of the areas where you put or cut holes. And if you want them to fruit, your hands, you need to create a little humid environment for them to live inside so that there's a ton of humidity so that they're triggered to fruit. Since I'm running so low on time, I just want to show you what these hands look like when they grow out. We can debut them together because I've been waiting for them to grow. So here's some hands. There's a little bit of fruiting action happening there. But once your glove is fully consumed, which this has been growing for 6 weeks, I think. So I'm gonna cut them open and bring them into. Are there any other last minute questions or comments or anything?
I think that's about it. But this looks, so they should be, if you're doing this yourself, you should be seeing kind of like slowly the mycelium will solidify and kind of expand, and then is that about
exactly
right?
Yes. So this is pretty hard. So they get really hard. They get really, really dense. And reishi is cool because, like I said, it's really rubbery. So it's sort of soft and bouncy. But yeah, you're letting it grow out for 6 weeks. You'll see it really start to kind of consume and grow into the substrate. And yeah, it takes the form of whatever it's in. So it's an incredible building material, an incredible queendom. And I hope that you'll come tonight to our performance that premieres at 7 p.m. I will talk more about it with my collaborators then. There's a lot of honestly, it's really beautiful and really mesmerizing, and it's an incredible project that's been cool to work on. And it's sort of a prototype as building this, working on hacking and building a sort of MIDI controller with the electrode sonification that I showed you by Merlin Sheldrake, because I'm actually going to start this experiment of the process of decoding and kind of trying to figure out the fungal language. So this project was sort of done as a prototype, but also to embellish these, this incredible phenomenon that happens between plants and fungi.
Oh, thank you very much, Kaitlin, for the wonderful lecture and workshop. And everyone give Kaitlin a hand. Well, I guess we'll be seeing you in an hour or two, so stay tuned on the YouTube channel. You can see the upcoming streams. And what was the name of the performance that you're going to be doing again?
Trans Relations at 7 p.m. Pacific time.
Great. So thank you again, Kaitlin, and enjoy. Thank you so much.
Okay. Thanks for coming. Bye, everyone.
Bye, everybody.